Hi, good morning. So thank you very much for the nice invitation. So thanks to the organizing committee. So I'm going to start to share my screen. So I'm from Lisbon, and so you are all welcome to visit us. We are a School of Engineering where I'm coordinating the biomedical engineer courses. So, and today I'm going to speak about work that we are conducting on my laboratory on biomarkers discovery for a precision medicine. So what is the precision medicine? Precision medicine means to a more effective medicine considering the difference between each individual. So the personalized concept of medicine and precision medicine is very similar, and considers that each person has a very specific way of respond, for instance, to therapeutic, has totally different mechanisms of disease and so on. For instance, when considering breast cancer, we have totally different types of breast cancer. So we need to understand the different types to adjust the therapy considering the type. For instance, if we are expressing hormonal receptors, the best therapy is going to be an hormonal therapy, and so on and so on. So, and these specific characteristics of each cancer, we could call it a biomarker. So for conducting a more efficient therapy, we need to discover these biomarkers. What is different between these different kind of looks like similar disease, but are not. So these biomarkers, so we characterize the different disease types and so on based on these biomarkers. We could use it to conduct a more precise diagnostic and consequently to define the best therapy, but also, for instance, to predict the outcome of the disease, so to conduct prognosis. And the best mode to discover these biomarkers is considering the whole of the system. So when considering the system as a whole, so we are considering all the genes, all the RNAs, all the proteins, or all the metabolites, we say that we are considering the omics science. So we are talking about genomics, transcriptomics, and so on. And during the last 20 years, technology has evolved a lot, so we can do lots of analytical characterization of these systems in a much cheaper and rapid mode. For instance, the first human genome that was sequenced and finished, actually was finished a few years ago, but almost 90% finished in the beginning of the millennium. It took lots of years, lots of monies around, the predictive value was around 3 billion to obtain the first human genome. Now, we managed to do a more or less sensitive analysis of a genome, of a human genome, in few days for a few $1,000. So it's much cheaper, much rapid. So this evolution means that most of the new first-in-class drugs, so drugs that are completely innovative in relation to the other drugs already in the market, most of them are already discovered based on these new technologies. So this is already a reality. But considering the omics science, we could speak about genes, but genes means that the potential of the cell is not really what the cell is doing. What the cell is doing in a particular system is producing proteins, and that are associated to the metabolism. So as we tend from the genotype to the phenotype, we could say that we're more near what the cell is really doing at this particular moment. So actually, I'm focused on at the end of the arrow, that is metabolites. So considering the whole molecular signature of the system, considering metabolites, but it's not easy to acquire this metabolome. We have different bottlenecks. One of them, the technique. What is the best technique to conduct? Actually, each technique are seeing a particular set of metabolites. So actually, we would need to use many different techniques, some of them more adjustable than the other ones. But even so, we are we are speaking about very laborious, very complex techniques and very expensive techniques. So, and these techniques also means that we are dealing with lots of information. And if you are associating this to a huge variability that happens in the human population, means at the end that is very difficult to discover biomarkers. So an alternative technique, the one that I'm working with, is with Fourier transform infrared spectroscopy. Actually, we are looking for the system as a whole, and we are looking for vibrations of functional groups and molecules. But this spectra that represents the molecular signature of the molecule at that defined period of time on a very specific condition is very sensitive to acquire the metabolic state of the system. So is a very nice and alternative technique. So we have been applied this technique to several systems, for instance, to quantitatively predict several metabolites, for instance, during the expansion of human stem cells. In this particular study, we evaluated 2 metabolites only, but we could do it lots of metabolites at once. For instance, in these systems, actually this one is a very simple system, like Escherichia coli bacteria producing a plasmid for vaccination. But it's a very good example how the technique could be using in a robust mode, because in this system we apply different types of E. coli in different types of conditions, and we predict a very a huge set of variables from a unique spectrum acquired in 1-2 minutes in a simple mode and very economic. Besides that information, and from the spectrum, we were also able to acquire lots of other metabolic information, like apparent transcription levels, the turnover metabolic profile of the cell, and so on and so on. So this is a very sensitive technique. A good example of the versatility of this technique is that we are able to develop a platform for drug discovery, actually work for antibiotics discovery. So, and I want to know if from a particular spectra, we were able to predict the mode that cell was being killed. So predicting the mechanism of death. So use different kinds of antibiotics. So we're killing from different modes of action. And actually, we were able to predict the mechanism of death of that cell. And even if the target of this antibiotic was different or not in the same class of antibiotics. So this high sensitivity of the technique is very, very appealing to discover biomarkers in the human population. One of the first examples that we applied for the human population is this one. Usually we are submitted to very genotoxic compounds, as on hospital, for instance, in professionals working the chemotherapy with chemotherapeutic drugs that are also carcinogenic. And usually this monitoring is based on conventional modes that require to take blood from the human volunteer or patient that we are monitoring, grow, expand cells or maintain the cells in culture during 1 week, and doing after very different analyses. This one that we are trying to evaluate chromosomal abnormalities in the immune cells on the blood. And this is very laborious. So to check if we have chromosomal abnormality. So in parallel to this analysis, we made another one that using this technique, this new one, and from a simple drop of blood, we needed to want to know if we're able to see subdivide this population, the one that were in risk and the other one that were not in risk. And just looking for the spectrum, we start to see a huge difference in very specific wave numbers. But applying more machine learning techniques as support vector machines analyzing the spectra, we were able to develop very nice prediction models of the genotoxicity associated to these particular populations with very nice prediction values. So with the advantage that it's very rapid to acquire this information. So this is a very nice example how the technique could be explored. A second example, what we did is there are many diseases that the characterizations of the cells, of the immune cells that are being activated is critical. For instance, in the transplant medicine, the mechanism of the immune reaction against transplanted organs is critical, and it's also critical to know the mechanisms, if it's a B or a T immune cell activation, and because in function of that, the therapeutics are different. And so on this particular work, we want to know if it were possible to discriminate these two types of immune cells, and we were able to discriminate it. This is already seen again on the spectra. Actually, this is the second derivative spectra. But again, applying machine learning models as the previous one, we're able to develop very good prediction models that we are able to discriminate it if we're talking about P or T cells, and more if these cells are being activated. So and predictions around 100% specificity and sensitivity, that is huge. The last type of examples before passing through a more complex analysis, we want to also from the blood, from the serum of transplanted patients, to predict if these transplanted patients were in risk of starting a rejection process of the allograft kidney that they added. So, and this is one of the results. So just showing that the prediction model just predict if the rejections were already starting was usual. We were able to predict the people that were already starting the rejection process and ones that apparently was not starting by conventional modes, but probably were going to be started on the future. That is a highly sensitive mode. And this is highly important, but particularly in this kind of patients, usually this is based on biopsy to the kidney, to the transplanted organ. That is highly invasive, and we are basing these prediction models on blood, so less invasive than using the biopsy analysis. So the last example is considering the tissue analysis. So before of that, all my examples were based on cells or on blood, serum, and so on. But sometimes on cancer, for instance, we want to analyze the tissue. So for the tissue, it's important to see the heterogeneity, and we never know if you are focused in the region associated to the cancer or not. So this is particularly difficult difficult analysis. So for this case, we use a total different technique based on a microscopic analysis, but with a very special detector that actually contains an array of detectors. Actually, it was 4,096 spectrums acquired, distributed in a very defined area. So we are speaking about imaging processing. The information is huge, so we need to apply different kind of processing techniques to simplify the information. Just an example, here we have the microscope observation of our sample without coloration, and you start to see the morphological observation. And these images are reconstructions from the infrared spectrum. For instance, example is integrating areas from the specific region of the spectrum. We're able to reconstruct almost the image at what you were observing here. Unfortunately, if you are looking for average of the spectrum for the normal of the derivative spectrum, we know that in average, they are different, but the standard deviations from the adenocarcinomas, that is cancer, and the tissue that don't have cancer and could be abnormal, like hyperplasia, that was our case in prostate cancer, and hyperplasia from prostate, they are superimposed. That so is not very good prediction. So we consider the image, this hyperspectra. Hyperspectra view has lots of spectra, and micro because we are using microscope imaging. So we evaluate pre-processing the spectra. We evaluate in reduction the dimension of the spectra to try to obtain more quick computer analysis. And we evaluate the non-supervised and supervised methods. Of course, the non-supervised didn't work because the variability associated is huge. But even so, we are able from the supervised classification to obtain accuracy in relation to the conventional classification of cancer, a huge one, 96%. So it was a fantastic result. I could say this kind of technique that is one of the most information-rich and concise way to represent whole omics, not only the metabolomics, but whole omics. So this is a very nice technique. So in conclusion, we could say, so this the technique has very appealing characteristics as rapid, economic, high throughput, and high sensitive. It captures the metabolic fingerprint of the technique and its associated metabolic state. And we are able to predict some biochemical information. And most important is all this information can be used for biomarkers discovery. So I would like to thank all my colleagues and students, and of course the grants associated to the Portuguese institution as Fundação para a Ciência. So thank you very much. So thank you again for the organizing committee. So if you have some questions, please be my guest. Thank you.